There's nothing I can use as a proof or something.
Good evening. Good evening.
Good evening. Good evening, madam.
So today we're going to talk about the immune system. I realize that your basic sciences are very weak. And so I've decided that for this particular lecture, we are going to discuss the basics of immunology. And then the next lecture, we will now be able to do the actual pathology. So today we'll just be looking at the basics, the cells, the components of the immune system and all that. Then tomorrow we'll look at the problems that can occur. I can't share my slide, Nick. Nick. Is Nick online? Hello?
Hello, madam. She's online.
Oh, man. I see his name there.
Hello,
But I
Nick.
can see him.
Yeah, I can also see him, but I can't share the slide. So I won't be able to. I don't know if he's physically present. Okay, whilst we wait, are there any questions before we move on? If there are any questions, let me answer them before we move on to today's lecture. I don't think it's around, so I still can't share my slides. Are there any questions? Okay. Then we'll just wait. When he comes, then I'll be able to share. Otherwise, we won't be able to proceed. I've sent him a message, but he's still not replied. So I don't know.
Yes, doctor.
Yes. Anytime.
Hi, Doc. Sorry.
Okay.
I had multiple. Okay. Doc, your account is okay now. You should be able to share slide.
Okay. I'm just answering a question and then we'll proceed.
Okay.
Okay, so today we are talking about the immune system. So we are going to discuss an overview of the immune system. So this is an overview. What it means is that it is a summary or a simplified form of what the immune system is and the various components and all that. So it isn't an exhaustive detail of immunology, but it is to give us a background so that we can understand. When we start dealing with the pathology of the immune system, then you actually understand when we say that somebody has a B cell defect or somebody has a T cell defect, you understand what exactly it means. And you understand, by understanding what the T cells do, you would understand when somebody has a T cell defect and how it affects the person. So the immune system classically is composed of two distinct interrelated components, and these are humoral immunity and cellular immunity. So the humoral immunity is non-specific. It is across board. It is mediated by soluble antibody proteins and then soluble proteins in the blood. The cellular immunity is mediated by cells, and typically these cells are lymphocytes, but you have other cells like macrophages, neutrophils, dendritic cells, and so on. Okay. So when we look at the immune system, basically, we have an acquired immunity and then innate immunity. So basically what this is trying to say is that the innate is inherent. Everybody will have this innate immunity, but the acquired one is from T cell or B cell. So if you take the innate immunity, there are two main things involved. Either there are physical barriers to infectious agents, which we looked at in a few previous lectures ago, where we talked about the skin, the mucous membranes, the saliva, the flushing action of urine and tears, such that they don't keep bacteria in the various organs. The stomach acid, the pancreatic, alkaline pancreatic juice, the bile, all those things are physical barriers. Then if you come to blood-borne members of this innate immunity, we have phagocytes and the complement cascade. So the complement cascade helps the antigen to be presented to the immune system for destruction by the phagocytes. And the phagocytes, we have neutrophils, macrophages, basophils, eosinophils, and natural killer cells. So these cells lead to the death of dangerous organisms, that is bacteria, viruses, fungus, and those things. Okay, now looking at the acquired immunity, it can either be T cell immunity or B cell immunity. So the T cell is where you have cell-mediated immunity, and the B cell is humoral or antibody immunity. And this acts by antigen exposure. So for B cell immunity to develop, you should have been exposed to the antigen. So for example, if you have what common organism?
Chickenpox.
Chickenpox, okay. So if you are exposed to chickenpox, it means that you have to be exposed to the varicella organism. Then when you are exposed to it, there will be lymphoblast formation. These lymphoblasts will produce plasma cells, which would produce the actual antibody against the varicella. And that is what will provide the immunity against the chickenpox. When you look at the cell-mediated immunity, it involves whole T cells. And the T cells can be either suppressor T cells, helper T cells, or cytotoxic T cells. And these would lead to the death of the microorganisms. So once you understand this diagram, you've got the whole immune system covered. Okay. And please, as I'm going on, if you have any questions, kindly ask so that I'll be able to answer as we go along. Okay. Okay. So this is a further explanation looking at the details of the cells. Okay. So the cellular components of the immune system are made up of the cells. Then we also, hey, what's wrong with you? Sorry. So we have the soluble mediators of the immune system. And those are the antibodies, the cytokines, the complements, inflammatory mediators, which help the immune system to be activated and to function. But they are not cells. They are proteins, and then they are mediators found in the plasma. When it comes to the cells themselves, we have phagocytes, and we have granulocytes and lymphocytes. And the lymphocytes, I already explained that we have B cells and T cells. And then we also have large granular lymphocytes. If you look at the phagocytes, there are mononuclear phagocytes, neutrophils, and eosinophils. And the granulocytes are made up of basophils and the mast cells. So each of these is activated depending on what happens. So let's take a few examples. Now, the mast cells and eosinophils are typically activated in an allergic reaction. Then neutrophils and mononuclear phagocytes typically for bacterial infections. Basophils would usually be triggered if you have parasitic infections or helminths. Okay, and eosinophils as well are also triggered by helminthic infections. Then the B cell, usually when there's antigen-antibody involvement, the B cell produces the requisite antibody. Then T cells help with killing of virus-infected cells and cancer-affected cells. Okay. The immune system also has organs, and these are collections of lymphoid tissue which help to produce lymphocytes. So we have the lymph nodes and the lymphatic vessels. That's why if you have a localized infection, you have the lymph node close to it getting swollen or enlarged, because you have the cells being rapidly produced there as a result of the infection, needing a rapid turnover of cells, immune cells, so that you can protect the body against whatever infection has invaded the body. So we have all these organs: the spleen, the Peyer's patches, lymph nodes, the appendix. The bone marrow is a storehouse of all these cells that we mentioned. Then the thymus is usually active in babies, in infants, in children below 1. Usually, if you take a chest X-ray and you see a central figure or a mediastinal figure, it tells you that the thymus is present, but it usually disappears with time. And so after infancy, you don't typically see it on the X-ray anymore. Okay, so I have sort of explained the function of the B lymphocyte. It neutralizes the microorganism either by activating complement or producing antibody. And then the antibody, so this is how the antibody acts. The antibody is produced to counteract the antigen. So you can see that the antigen has specific structures on it in the various shapes that are shown on my right. So the antibody has a complementary shape so that it attaches to this and helps with the neutralization of the antigen. And so the antibodies that are produced are specific to the particular infection that has invaded the body. This is different from the innate immune system, which is general. So if you take the skin, for example, the skin acts as a barrier to all microorganisms. It is not specific to viruses and then maybe allows bacteria, but then it prevents viruses. It prevents entry of all microorganisms. But if you take the humoral immunity regarding the antibody production, it is specific to the particular infection. Okay. Now, the T lymphocytes, usually what they do is that they lyse the target cells. So lysis means breakdown. So they cause breakdown of the target cells. And they do this by causing the production of soluble protein mediators, which are called cytokines. We saw cytokines when we talked about the inflammatory process. And here too, we see cytokines. And the cytokines are mainly produced by the T lymphocytes. So when the bacteria enters the body and the T cells are stimulated, they act by producing cytokines, and the cytokines would cause whatever reaction to go on to help with the killing of the microorganism. Now, the T cells can only act on the antigen when they recognize certain things on them called major histocompatibility molecules. So if they have the major histocompatibility molecules, then they can engage the T cells. And so for short, it's called MHC. So the MHC, we have MHC class 1 and MHC class 2. We'll look at this further into detail. Now, we also have natural killer cells. So natural killer cells are large lymphocytes. And what they specifically do is that they kill cancer-infected cells and virus-infected cells. Okay. Now, each T cell is also genetically programmed to recognize a specific T cell receptor. So similar to what happens with the antibody response, the antibody recognizes a specific antigen. So in the same way, the T cell too has a T cell receptor which recognizes a particular antigen. Okay. So let's look into detail at the T cell receptor. So in most of the T cell receptors, it is a molecule composed of alpha and beta chains. Now, each of these alpha and beta chains, they have a variable. Variable region is what is changed depending on the specific target molecule that you are looking at. And then the constant region is what is responsible for the intracellular signaling. So that is constant for all the lymphocytes. So this T cell receptor recognizes non-protein molecules. It recognizes bacterial cell walls. It recognizes viral cell walls. And the T cells also express other molecules. And typically, two types are known. We have CD4 and CD8. And if you look at the ratio between the CD4 and the CD8, it's about 2:1. So we have twice as many CD4 cells as CD8 cells. And then interestingly, the CD8 cells would recognize and bind to class 1 MHC molecules or class 1 major histocompatibility molecules. And then the CD8 is on about 30% of T cells. So the CD4 recognizes MHC2 and then CD8 recognizes The MHC1 recognizes it. Go and sit down and drink your water. The CD8 recognizes MHC type 1 molecules, and then the CD4 recognizes MHC type 2. So I remember when I was in school, it used to confuse me. So what I told myself was that whatever the product is, you get 8. So if it is 8 times 1 is 8, and then 4 times 2 is 8 as well. So the CD4 cells they recognize MHC2. That's 2 molecules, and the CD8 recognize MHC1 molecule. That's how I try to remember. But as time went on, you remember these things because of the various applications that you put them into. So the different CD4 and CD8 cells, what do they do? The CD4 cells are T helper cells. T helper means that they themselves do not physically kill the microorganism. They produce cytokines, which help to produce antibodies, which would do the killing. The CD8 cells are cytotoxic. They themselves will do the killing. Okay, so CD4 cells are helper. CD8 cells are cytotoxic. They will kill themselves. Okay. Then B lymphocytes are from the bone marrow, and that's why they are called B lymphocytes. And they form about 20% of the population of lymphocytes in the blood. You usually find them in lymphoid tissue, so lymph nodes, spleen, tonsils, Peyer's patches in the GIT, appendix in the GIT, and then the spleen. And they are the ones that produce the antibodies. So we know that there are 5 types of immunoglobulin antibodies: IgG, IgM, IgA, IgE, and IgD. The majority of the antibodies are IgG, IgM, and IgA. Let me just talk a little about each of them. The IgA is responsible for mucosal protection. So if you have a deficiency of IgA, you have problems with your mucous membrane, so the respiratory tract, the GIT. So things like people who have constant colds, people who have diarrhea with a listing, there might be a problem with their IgA. Then we also have IgG. IgG is the main immunoglobulin class. And we also have IgM. IgM is usually secreted at the initial phase of the infection. Then later on, IgG takes over. However, there are small levels of IgE and IgD. The function of IgE is in immune hypersensitivity reactions or allergic reactions. That's where you find IgE. IgD, basically, it doesn't really perform much of a function in the immune system. Akosia, pardon no fear. Leave the bed. Come and sit down. Hurry up. So, yes. So, just like the T cell receptors, B cells also have receptors. And the B cell receptor, it is unique for each antigen because it helps with antibody production. And so it is peculiar. Okay, yes. It is peculiar to the particular antigen that it faces. So I've summarized the differences between the two. So B lymphocytes, usually they mature in the bone marrow, while the T lymphocytes, like the name suggests, it matures in the thymus gland. Then B lymphocytes is just about 20% of the total blood volume of lymphocytes, and then T lymphocytes form 80%. The B lymphocytes are usually involved in the humoral immune response, whilst the T cells are usually in the cell-mediated immune response. The B cells, the mature B cells, usually you find them outside the lymph nodes, but then the mature T cells are usually found within the lymph nodes. And if you take the B cells, the subpopulation, what this means is that at any point in time, if you have antigens being exposed, you have two types of B cells, either memory cells or plasma cells. The plasma cells are the ones that develop into the antibodies. The memory cells are the cells that are there with the immunologic memory, so that you will not have that same infection twice, because there will be immunologic memory. So if you encounter that same pathogen again, the antibodies are readily available and they will control the infection. Okay. Now, when you take the T lymphocytes, of course, the subpopulations are the cytotoxic T cells, which is the CD8, and then the helper T cells, which is the CD4. Okay. Now we look at macrophages. So macrophages have the MHC class 2. What this means is that they can bind to the CD4 cells. If you remember, I was saying that the product of the MHC and then the T cell should be 8. So the CD4 cells, T cells, would recognize the MHC class 2 molecule. A simple way to remember it is that, you see, if you have T lymphocytes, they have their specific cells that they target. So for a T lymphocyte, which is of the CD4 type, it looks for cells which have the MHC2 on them. Once that recognition is there, they can work together. Then the cytotoxic T cells, the cytotoxic T cells, they go alone, and so they go alone and they target cells with MHC class 1 on their surface. Okay, and what do macrophages do? They produce cytokines. We'll eventually look at cytokines and we'll understand a lot more about what they do. And then these cytokines would influence the T cells and B cells to produce other cell types like endothelium and fibroblasts. Then the macrophages themselves can also phagocytose, so they can actually engulf the bacteria and kill them. Okay. Apart from macrophages, we also have dendritic cells, and these are cells of the immune system which have a dendritic morphology. If you remember the dendritic morphology from is it physiology or cell biology, they have these processes like the nerve endings which synapse with other cells, and usually there are two functional types. We have the interdigitating dendritic cells and the follicular dendritic cells. So the interdigitating ones, they are non-phagocytic, so they do not phagocytose the cells, and usually you find them in lymphoid tissues and then non-lymphoid organs like the heart and the lungs. If you find them in the skin, they are called Langerhans cells. Then the follicular dendritic cells, usually you find these ones in the lymph nodes. And if you take a section through the lymph node, there's something we call the germinal center. That's where you usually find them. Okay. And they produce the Fc region for immunoglobulin. So they are sort of like antibodies. If there's an antigen, there's that antibody response from the follicular dendritic cells. Okay. Then we have natural killer cells. Please hold on. Okay, so the natural killer cells, they kill cancer-infected cells and viruses. And they have, this killing is done by the presence. We've encountered this as eosinophilic granules before, when we were talking about inflammation, when we're talking about the final stage of the killing of the bacteria. We said that a lot of the neutrophils contain azurophilic granules, which bind to the membrane of the bacteria, and then they release their toxic contents containing enzymes to digest the bacteria. So here too, natural killer cells have these azurophilic granules. And they also have some molecules on their surface called CD16 and CD56. They also have the capacity to cause antibody production, and this is called antibody-dependent cell-mediated cytotoxicity. And they also secrete cytokines, and typically the cytokine that they secrete is interferon gamma. Okay, so this is how the natural killer cells work. The natural killer cells usually they recognize the MHC type 1 in the cell. So every healthy cell, you realize that the healthy cell has MHC1 receptors on it. Whenever the healthy cell becomes infected with a virus, it will lose its MHC on the surface, and that will make it a target to be destroyed by the natural killer cells. Okay, so that's what usually happens, and then the natural killer cell will cause breakdown of the cell. Okay, so we sort of talked about this earlier, that the MHC or the major histocompatibility molecules are of 3 categories: class 1, class 2, and class 3. The class 1 is linked to human leukocyte antigen. So this is this is genetics as well. So the human leukocyte antigens, there are 3 loci on the gene chromosome 15. We have A, B, and C, and this is recognized by the MHC class 1 molecules. And the MHC class 1 molecules interact with CD8 cells, which are the cytotoxic lymphocytes. Because MHC type 1 is present on virtually all nucleated cells, if you have a virally infected cell, that cytotoxic T cell is able to recognize it because it will lose its MHC type 1 and it will become a target for killing by the lymphocytes. Okay. Now, the MHC class 2 molecules usually they are coded by genes in the HLA-D region, so D, P, Q, and R. And usually, this is found on monocytes, CD4 T lymphocyte interacts with monocytes, macrophages, dendritic cells, and B cells. And all these are capable of producing antibody. Okay. So the MHC type class 3 is usually the complement component. Okay. And then also some cytokines. So tumor necrosis factor beta, tumor necrosis factor alpha. So usually you don't consider the class 3 unless you're going to do a transplant. Once you are thinking of transplantation, then you'd have to match all these 3 histocompatibility types. Okay. So what is the significance of the MHC molecule? So usually an individual will inherit 1 HLA allele from each parent. And if the person is heterozygous, it means that they will have 6 different HLA molecule of paternal origin. And then, so that is 3 of maternal origin, 3 from the father. So that makes it 6. Because each allele is 2 loci. So usually, this is what the significance is. Now, if you are going to transplant an organ, you have to match the HLA of the person who is giving the organ against the person who is receiving the organ. If the typing does not go well, or if they don't match, then what will happen is that the host or the person receiving the organ would form transplanted, and there will be a graft versus host reaction. And so usually when we transplant, we test. Before the transplantation, testing is done. Now, after the transplantation, patient is put on immunosuppressive drugs to sort of quieten the immune system so that the person will not reject the new organ that has been transplanted. Now, there are some diseases which are also associated with some HLA, selected HLA types. What I'm trying to tell you with this slide is that every disease has a genetic base, or most diseases, or I shouldn't say every diseases, many diseases have a genetic basis, or people have genetic susceptibility to diseases. Okay, so if you have this HLA-DR3, you are prone to chronic active hepatitis. So let me just explain further. For hepatitis B, there are 3 scenarios. Your body can either overcome the infection, you clear the infection, or you become a chronic carrier, or you progress to hepatocellular carcinoma. So if you have the DR3 gene, then you end up with a chronic active hepatitis. Also, there's this insulin-dependent diabetes mellitus. When you have HLA, DR4, DR3, that predisposes you to diabetes. So these are some of the reasons why certain infections are familial. Okay. So finally, we'll end with a closer look at cytokines. So what are cytokines? Cytokines are just proteins that are in the blood. They are low molecular weight polypeptides or proteins, and they are produced by cells in the body: macrophages, monocytes, neutrophils, and so on. And what do? They they are mediators. So they help the cell to achieve its function. And there are 3 ways by which they do this. Either by an autocrine effect. We looked at intracellular signaling. And if we say autocrine effect, what we mean is that this cell will produce the lymphocyte or the cytokine. The cell will produce the cytokine, which will act on the same cell to help to propagate whatever is needed, if it's growth, for example. Then we also have a paracrine effect, whereby the lymphocyte or the cytokine will produce an enzyme or a chemical which will act on an adjacent cell to cause a change. So a change can be differentiation, a change can be chemotaxis, a change can be inflammation. Then they also act by endocrine effects. And the best category is the production of interleukin-1 and tumor necrosis factor, which would be involved in the inflammatory process. So what are some of the things that cytokines do? They act in amplifying cascades. Amplification of a small cytokine that is released. We see this example in septic shock, the pathogen